So meiosis begins with interphase. So this represents the nuclear envelope, and we're going to draw some centrioles up here. And there's going to be replicated DNA, so they're going to join at the centromere to become sister chromatids. So it's going to be like this. And the next phase is called prophase 1. So this is when this nuclear membrane, take that off. It's hard to do with one hand. Right. And then synapsis is going to occur. So this means that these chromosomes are homologous, and they're going to start moving closer together, and they're going to pair up and join together. And this group of 4 different chromatids is called a tetrad. So the next thing that's going to happen is the centrioles are going to move to the opposite sides of the cell, and this entire board represents the cell membrane. So after they move to the opposite sides, the next step that occurs is called crossing over. So I'm going to redraw one of these so it makes it a little bit easier to tell. So this is when the DNA is going to break off of one of these chromatids and rejoin on the other, so they're going to exchange DNA. It's going to kind of look like that. And this would not happen in the prophase of mitosis. So the next step is metaphase 1. So this is when the chromosomes are going to move in a line across the center of the cell in homologous pairs. So I'm going to rearrange these. It's kind of going to be like this. So they're going to be, in all actuality, they would be much closer, and there would be more on either side of them. I'm just going to use these 2 to represent. So in metaphase and mitosis, they would just have a single line of sister chromatids, not of the homologous pairs. So the next phase is called anaphase 1, and these homologous chromosomes are going to separate and move to the opposite sides of the cell using spindle fibers. So that's going to look like. It's going to kind of be like this. So kind of be maybe start being pulled that way. And then these spindles here, they're going to attach to the cells at their centromere, which is right here, and that's going to be called the kinetochore is the structure that's going to attach to these centromeres of each chromosome. But in the anaphase of mitosis, only single chromosomes would pull apart, not the entire homologous chromosome like it is here. So the next step is called telophase 1, and this is when cell division occurs, and the centrioles are going to replicate. So this is going to result in 2 daughter cells. So that line down the middle is going to make 2 different cells, and there's going to be a sister chromatid in each one. And the centrioles are going to, and then the sister chromatids will differ from the chromatids of the parent cell because the genetic info was exchanged during crossing over. So the next thing we're going to do is draw a nuclear membrane, and then these centrioles are going to duplicate. So this would take place in interphase during meiosis 1. So the next step, or the next half of meiosis, is called meiosis 2. So this begins with prophase 2. So this is when the centrioles are going to move to the opposite poles of the chromosome. So they're going to go from here to here. And these centromeres are supposed to or these chromosomes, sorry, are supposed to move towards like the center of the cell, but at the same time I'm just going to leave them like that and imagine that's happening. So this is different from prophase 1 of meiosis because the two haploid cells are formed. So so I'm going to form this, so it can make more sense when explaining the next steps, add some extra chromosomes. So this next step is metaphase 2. So these centrosomes are going to line up in a single file line in the center of the cell. So it's going to look like this. So these centrosomes are going to line up. And this is different from metaphase 1 because instead of the homologous chromosomes lining up, it's just the sister chromatids. There's not another pair like right here. So the next step that's going to occur is anaphase 2. And these chromatids are now going to be drawn to opposite sides of the cell. So these sister chromatids are going to split in half and be pulled to the other sides of the cell. And same thing by spindle fibers. And this is when, so these chromatids are being split into just chromosomes. So the next step that's going to occur is telophase 2 and cytokinesis. So this is when the cell division occurs and is completed. So it's going to basically just kind of be like this. It's a little messy, but... So 4 daughter cells are going to be formed out of these 2 parent cells, and I believe I included all the vocabulary. The only one I didn't mention was the synaptonymal complex, and that's when during prophase 1 duplicated homologous chromosomes line up and physically connect along their lengths by a zipper-like protein called synaptonymal complex. So I believe I included everything else, and thank you.